Can everybody see that all right?
Yeah, I can see that.
Perfect, and the pointer as well. Perfect. Right then. So just an overview to start with of what I'm going to talk about today. So to start with, I'll go through kind of a general understanding of the general concepts and aims of phase 1 trials. Then I'll move on to some solutions to trial design in terms of traditional 3+3 design, talk through the continual reassessment method, which is another trial design that we use for utilised for MUK-11, and then more recently kind of a model assisted design with ProMMise. And then I'll finish up just by briefly introducing some of the ideas in my PhD, looking at both toxicity and efficacy-based dose finding. So the drug development pathway as an introduction, obviously this is phase 1, what we're talking about today. So clinical research develops in stages. We start within a lab model, move towards animals, and then start to answer kind of clinical questions in a staged way. So phase 1, we'll talk about safety, tolerability, and dosing, and looking at kind of 10 or 20 patients, and then move to phase 2 and phase 3. But most of you on the call will be aware of this. So we're talking about phase 1 and dosing and safety. Okay. So the general aims of phase 1 trials are to find the maximum tolerated dose or the recommended phase 2 dose. So there's a general assumption here that more treatment is more efficacious. So we want to treat at a dose that's high enough that gives acceptable toxicity profile, but then also by treating a higher dose, we're also assuming that we've got more efficacy. And our basic endpoint to do this is based on dose-limiting toxicities, and we assess this through a DLT window. So we're talking about serious toxicities that will limit treatment, and we'll look at these over, say, 1 cycle or a 4-week cycle or an 8-week cycle. And another aim of that is to understand tolerability and safety, and this could also then include kind of efficacy or other outcomes in a dose expansion type stage. So then looking specifically at kind of dose-limiting toxicities, these are side effects that prevent the treatment being given at a higher dose, and these are predefined in the protocol according to specific criteria, and these are within a specific time frame. So an example, DLTs will be assessed during the first treatment cycle up to the administration of cycle 2, day 1, and must be toxicities considered related to the drug. And we define these as absolute neutrophil counts, for example, platelets less than a particular limit and lasting more than 7 days, and then more than a 3-week treatment suspension during cycle 1 due to toxicity related to the drug in question. So then a couple of types, or 3 types of designs that we might utilise to answer these questions are the rule-based designs, which some of you will be familiar with, such as the 3+3. And then we'll talk a bit more around the model-based ones. So continual reassessment, Bayesian logistic regression, and time to event, if for a slightly different endpoint. And then more recently, kind of some model-assisted designs that we've utilised for ProMMise. So the simplest design, and has been around from kind of the 40s, I think, 40s or 50s, is the 3+3 design. So these are referred to as kind of a rule-based design. So the trial will start, we'll define a set number of dose levels, and then we'll usually start at the lowest dose level. We'll enter 3 patients into the trial, and then we'll see, observe over that DLT window how many DLTs we've seen. So if we've got less than 1 DLT or no DLTs, we'll escalate to the next dose. If we've got 1, we'll add a few more patients because we're unsure. We'll add 3 more patients and then look at the rule at 6. And then if we've got 2 or 3, we'll de-escalate because the dose is too toxic. So we'll move around the kind of dose space until we find the MTD, and then we'll declare at the end of the trial, the MTD is usually the dose level or the highest dose level with less than 2 out of 6 DLTs. So obviously this is quite simplistic. You can write it in the protocol, is probably one of the positives of it. But beyond that, from a statistical perspective, it doesn't work very well. It doesn't find the purpose of the trial, or the aims of the trials, particularly well. So it will often undertreat in terms of it takes a long time to get towards the MTD. So obviously with patients that are on trial, they seek a therapeutic benefit. So we want to treat as many patients as possible there or thereabouts the MTD. And we find with the 3+3 design, what happens is it takes a long time to find the MTD, and we're not particularly reliable at finding that MTD at the end of the trial. So as a proposed solution, back in the 90s, John Quigley proposed what's called a continual reassessment method. So could we utilise statistics to better solve the design problem? And this is still, despite it being quite superior in many ways, it's still not popular. I think it's around 90% of trials will still use the 3+3. So I'll quickly introduce this now. The aim is to find the dose with a pre-specified target toxicity, the target toxicity level. So this is the probability. So we could think of it as a long-term kind of frequency. We expect around 30% in this example from MUK11 of a reassessment drug. We want approximately 30% of patients to have a DLT, and that will, while that's seen as a compromise between maximising efficacy and kind of maximising or limiting the amount of toxicity. We agreed that kind of we're targeting around 30% of participants to experience a DLT. And so, yeah, so the next one. So then the general concept really is that a dose toxicity curve is assumed at the design stage. So we'll think about linking the probability of toxicity at any given dose, and we'll come up with kind of a proposed solution as to what that looks like. I'll have a figure next for you to visualize in the next slide, and then that's based on clinical expertise, preclinical data and other trial data. And then after each patient's data is observed, the curve is updated, and that's based again on the number of DLTs out of the number of patients. And then the next dose is chosen as the dose with probability of toxicity closest to that target toxicity. And then the trial finishes, unlike the 3+3, after a set number of patients have been recruited. So thinking about the MUK11 trial, we specified that after 22 patients, there was 4 dose levels, and after 22 patients, we'd make a decision as to which one was closest to this 30%. So I mentioned this kind of dose toxicity curve. So here we've got dose level, and in this dummy trial, we've got 6 dose levels, and then along the y-axis, we've got the probability of toxicity. So we see that as a general assumption that as we increase the dose level, that the rate of DLTs increases as well. So this is based on preclinical information as a general idea as to which dose we believe to be the MTD. So here we see the closest dose level is dose level 4. We see that that's of this 30% target toxicity, got 5 below 35 and slightly nearer than 20%. So then we recruit a single patient, and we recruit them at dose level 4. And then we see that there's no dose-limiting toxicity. So we can see that the curve is estimated to come down from the original prior. So we then see that the dose that's closest to this target toxicity of 30% is dose level 5. So we treat the next patient at dose level 5. And then dose level 5, we see that that patient has no DLT, and the curve is updated, and we re-estimate and treat until the 22 patients have completed. Now, obviously, there's a slight danger with this, that this can baffle some people, and it's almost this black box approach where you put in numbers into an algorithm almost, and out of it spits a dosing decision that's not particularly well understood. So it's to stress really that this dose decision at any given stage is a combination of both statistics and then additional kind of information. So the trial will proceed in terms of after each dose or patient has been treated, we'll then look with a panel of clinicians and treating clinicians, and then look at additional data such as kind of lesser grade toxicities, and then also kind of perhaps the comorbidities or whatever of the patient. And so we'll really input this clinical expertise, and it's important to stress that the clinical team can overrule kind of what the model is suggesting. And so then this proceeds. We have a panel that will discuss the DLTs in question after each decision point, and then at the end of the trial we'll then have an MTD that's recommended for phase 2. So the CRM that I've talked about in terms of the example was the first trial that was, I suppose, proposed, and there's a few factors that didn't quite match with what was expected in terms of when we took that to practice. There's a few basic modifications that can be easily made to the CRM or criticisms. So that one of them is that we start at the lowest dose. We looked at the example, perhaps it's unethical to start at dose level 3 with little information. We might want to treat in cohorts, and the main advantage of this is that it speeds recruitment up, which I'll come on to talk on the next slide around ProMMise. Another one is that you don't skip dose in escalation. Obviously, the dose toxicity curve can move quite a bit just on little information. We don't want to go from dose level 3, for example, all the way up to dose level 5 if we've not explored dose level 4. So we can stipulate that these ad hoc rules, so we don't skip a dose in escalation, for example. Another thing is I'll talk about in ProMMise again, we can stipulate early stopping rules. So these are to prevent patients being exposed to overly toxic doses, as well as to reduce that sample size of 22 that we talked about for MUK-11. And then quickly, you can also kind of have different escalation type procedures. So you can allow intermediate doses. So we can skip doses in escalation, and then we might see, and then in other trials, we have seen that there's intermediate doses in de-escalation. So you might get somewhere around the MTD and then explore a dose similar. The ProMMise. So this is a platform trial for relapsed patients to evaluate ongoing novel therapies in multiple myeloma in combination with standard of care therapy. So Rakesh talked at the last presentation in detail of this, so I won't go too much into the clinical stuff, but I'll mainly highlight this kind of part of the design, this stop-go kind of point, which is kind of a phase 1 design, and talk about the design of the phase 1 component. I did just want to mention the other bit of the statistics, really, that the advantage of the platform really is that you can really speed up the development of the phase 1 trials. So what happens normally is you'd have this cohort of 3 patients. You'd then stop to wait for every patient to finish their kind of DLT window. And when we originally proposed ProMMise, it was going to be a 12-week observation period. So you can imagine if you recruit 3 patients and then you have to wait 12 weeks, it kind of limits the amount of data that you can collect within a year. So what we've proposed here is kind of a flip-flop design. So the monotherapy arm will go straight into cohort expansion, and then so if there's an arm open in escalation, we'll prioritize patients getting access to that, and then the arm will shut, and then we'll flip towards the monotherapy, for example. So we kind of flip between each of the arms to maintain constant recruitment, and this really brings down the length of the trial, the total length of the trial, to bring novel drugs in combination to patients quicker. Okay, so the basic premise of this design from ProMMise was there was a number of different arms that had slightly different questions. So some, the cyclophosphamide arm, for example, it was, can we safely deliver the dose of belamaf with cyclophosphamide? If not, we might want to jump down an arm, so it's almost safety running. Whereas for the belamaf plus the PI, it was a more formal dose finding. So I think we had 3 dose levels for this arm, and we want to start at the lowest and then formally find. So we want a design that was flexible, knowing that at any given stage we might introduce a new arm, and so we wanted something that was quite flexible and easy to run along the trial. So The model-assisted designs have been around for about 5-6 years now and came after kind of the CRM. And there's no assumed relationship here. It's a simplification of kind of the model-based designs. And we, rather than modeling kind of that dose toxicity relationship across doses, we're just modeling the probability of observing a DLT at any given dose, and then looking to model the kind of decision criteria. So if we think by definition, the probability is between 0 and 1. So 0 would be there's no chance of a DLT, it's impossible, whereas 1, it's certainly going to happen. So we split this region into 3 different regions, or split this domain into 3 different regions. So if we're in the 0 to 0.2, we're saying that it's quite low and we might want to consider a dose that's higher. Between 0.2 and 0.3 is just about right. So we should explore or treat more patients in that range, or treat doses, or stay at the same dose. And then if we're in this higher region, we're saying that the dose is a little bit too toxic, so we should de-escalate down and look at to see whether that dose below is more appropriate. So real advantage, this is taken from the ProMMise protocol, is that we can pre-specify the decisions in the protocol. So with the CRM, you have to, after each cohort, you have to rerun the model and include that in a safety review report. Whereas in this stage, we can put a table into the protocol, and so the decisions are made up front. So this table on the top, we've got the number of valuable patients. So this is the number of patients that have been treated, and then the number of DLTs that have been observed for each of those patients. So we just simply, at the stage of the SRC, just look to see how many patients there were and how many DLTs. So in this instance, if there were no DLTs in 3, we'd escalate, sorry, 1 out of 3 would stay the same. And you can see there's slightly different decisions at each of 3, 6, 9, and 12. I think this table is not complete, so we can also use this design to, if, for example, we have 4 patients, we can still make the decision as to which dose to use. So then, thinking about stopping rules, there's two basic stopping rules. We've got one would be for a patient benefit. So if we see that a dose is too toxic, we obviously don't want to treat at that dose again. So this will be the red criteria here from the stopping rule will be don't treat at this dose again. And this is based on from this slide here. We've stipulated that anywhere between this region between 0.2 and 0.34 is about correct dosing. So we've made our decision as to whether the dose is safe or not, as to whether it's below this threshold or above in the purple region. And this corresponds to the probability. So if there's a really low chance of it being below 0.34, then we don't want to treat there. So that in these situations, we have 2 out of 3 or 4 out of 6, we won't treat at that dose again. So the trial proceeds similar to the 3+3 design, I suppose, where you will treat, look, and then either escalate or de-escalate. And then the trial will stop when you've either reached the stopping rules or maximum sample size that's a little bit lower. So then, I briefly mentioned before, kind of that the 3+3 wasn't very good. So I just wanted to summarize really, I've lumped model-based and assisted together, and I'll talk about kind of the disadvantages of these at the end, or the difference between the two. But really, compared to the 3+3, there's a really clear loser. So we look at each of the criteria from the aims of the phase 1 trial, and then we look how well this is met by both the rule-based, which is the 3+3, or kind of our model-based type designs. The target DLT rate doesn't really exist in the 3+3, and we actually find that the maximum tolerated dose is entirely dependent on the number of doses that we treat or propose. So if there's a large number of doses, where it's very likely that we'll select kind of the MTD quite low down, whereas for a low number, it's less likely. Whereas with the model-based, it's clearly defined and can be flexibly chosen. So different situations require a slightly different trial design, so it's super flexible. Patients treated at the optimal dose, I mentioned before that the 3+3 takes a long time to get up to the MTD, and then once it's there, it tends to treat relatively few patients. In contrast to the model-based, which will get up to the MTD quite quickly and then treat many patients optimally. The main reason for this is that it doesn't use a lot of the data that's available. See, it's just using that initial cohort of 3 patients, whereas a model-based and model-assisted will use all the information at that dose level, or in the case of the model-based design, across different dose levels. And then it's also not particularly flexible. If we think about slightly more complicated questions, it's difficult and quite dubious to how to do that in the rule-based setting, whereas in the model-based, it's smooth and relatively straightforward to answer different questions and to slightly adapt the design. So one of the main benefits of kind of model-assisted designs, though, is the deviations from the plan. So what can happen is that for whatever reason, we're at the SRC meeting, we've only got information for 5 patients, for example. In the model-assisted and the model-based designs, we can easily kind of compute a decision for the next group of patients, whereas in the rule-based, there's no real rule. And then similar, I was sat on a safety review committee for a different trial, and there was information after a while that came that a DLT that wasn't reported, so we made the decision to escalate, and then that was then declared a DLT. And it's what you do in those situations, because it's not clearly defined. In the model-based, we can just plug those numbers into the model and then make an appropriate decision, whereas in the rule-based, it's a little bit more complicated. Okay. So I did mention that I'd talk briefly around the model-based versus model-assisted. I think this really depends. It's not a simple yes, one is better than another. It'll depend on the number of dose levels that you're studying, kind of the exact question that's at hand. So I think in general, I would be using kind of a model-assisted type design if there's a few dose levels, whereas if there's more dose levels, I think I'd consider more the model-based. And a lot of this is around practicalities as well as kind of the statistical properties. Okay. So I mentioned at the start that there is this assumption that more toxicity or higher dose is improved efficacy. So here, this is a curve on this kind of x-axis. We've got the dose, so increasing from left to right, we've got increased dose, and then this is just a measure of effect. So we've seen with what we've talked about so far, that as we increase the dose, the toxicity increases with dose, and then ideally, with these designs, we want to treat at the MTD. And there's an underlying assumption efficacy kind of broadly kind of increase at a similar rate to the toxicity in these types of designs. Whereas we can see with this curve that there is a little bit of a plateau. So quite a simplistic example, a recent example from multiple myeloma would be the belamaf compound that we're using in the ProMMise trial, where the recommended phase 2 dose from the DREAM-1 study was 3.4 milligrams per kilo. Then they looked in the DREAM-2 study at 2.5 milligrams and a 3.4, comparing, and found that there was little difference in the efficacy, but then one dose, the higher dose, was more toxic. They've gone, there's quite a bit of scheduling differences, but they've gone with the lower dose, and this is probably an example of this type of scenario where you'd want to consider looking at both the efficacy and the toxicity at the same time. We see that for quite a big improvement in dose, there's very little increase in efficacy. So in this scenario, we might think that this dose here is the optimal rather than the MTD. So we want to undertreat in terms of toxicity, but it's a very similar efficacy level. And this is more common now with some of the different classes of compounds, unlike the cytotoxic agents. So I think that's a really fast whistle-stop tour of phase 1 designs. So I think just in summary, don't use A+B trial designs in phase 1 for the reasons that they don't treat, they're not very good for patients and they're not very good from a scientific perspective. And evolve a statistician when you can, and underlying really is that efficient and novel trial design in phase 1 could treat more patients optimally, in addition to obtaining more robust answers to the scientific question at hand. I think there is an assumption sometimes just to get phase 1 out of the way, to get to the more exciting kind of efficacy and get it into the clinic. But actually, by spending your time at phase 1, we can have more successful, or phase 1, phase 2, more successful phase 3 trials by optimizing the trial design of phase 1. And that brings me to the end. Any questions or comments?
Thank you, Andrew. That was a really nice summary of the designs, and I'm really pleased to see the third bullet point that you've got on there, because as you were talking, I was sort of typing out some thoughts and actually really pulled out that point that efficiency is not. Sometimes we think about efficiency in speeding through a trial, so as few patients as we can to get to the answer really quickly. But actually, we need to think about our phase 1 trials as being part of the whole drug development process. And it's really the start of clinical trials research. So we need to make sure that the answers that we get in those starting trials are robust and reliable to inform the later practice changing trials. I think that's a really, for me, that's a really sort of key take-home message, to not just think about efficiency in terms of small numbers, think of efficiency over the whole drug development pathway. Does anybody have any questions?
Yes.
Oh, Lisa, I can see your hand. Yeah.
Yes, I have one. Whilst I can well, first of all, thank you very much. It was extremely informative from a patient's point of view. So you'll understand that I'm not that au fait with anything that any sort of thing that you do. But it did cross my mind, whilst it might not be possible to do it with what you're doing, anywhere in any of the designs, is there a variable for gender?
So I think it's within multiple myeloma, I'd say that the drugs don't tend to have a kind of gender component, but obviously with other drugs that can be designed, these are adaptable, particularly the model-based designs. If we think that the MTD may be different between not just gender, but any kind of subgroup, so these biomarker designs particularly might think not just gender, but lots of other characteristics that might infer a different MTD. So there's lots of research now to look at, is the MTD the appropriate to give all patients, or is it actually more personalized medicine where we're looking at different subgroups and even kind of individualized maximum kind of tolerated doses? So in other trial areas, they're looking, for example, to see, starting off low to see how patients respond to the first cycle, and then we'll make a decision whether to escalate for the second cycle, for example. But it's really, I think what I've proposed is the more simplistic one MTD for the entire population. But certainly there's lots of different subgroups, and it just depends on the situation and yeah, the setting, I suppose.
So where would that come then? Phase 2, phase 3, or?
So it would generally come, if it was referring to the dosing, it would come at the phase 1, phase 2 stage. So if it was responding more to the efficacy, so if, I don't know whether I'm still sharing my slides. So
yes,
if we go,
you are.
I'll go back to this one. So if we look here, it will depend on the question, the exact question. But really, if it was concerning more efficacy, we'd refer to it as phase 2. Whereas if it was more the safety around the dosing, it would be phase 1. So it's important right at the start, as Sarah mentioned, to kind of answer these questions that are really important for, I guess, getting the compound or whatever approved, is that you answer these questions early on, so that you don't end up in the phase 3 setting where you find out that there is a huge biomarker or gender-based component that basically means that your drug's not going to be approved.
Yeah, I would add to that as well that although it looks, when we display these figures, it looks like it's a very linear process from one study in the lab to a phase 3 trial, but it could very well be that actually we undertake a phase 1 trial on, let's say, all comers, and then we start to see a signal in one population or another. So we might do an expanded phase 1 trial in one population whilst we move to phase 2 in another population. And sometimes it can sort of, we can do a phase 2 and then need to go back and look at safety in other populations. So it's this sort of multiple pathways that it can take when we're evaluating different doses.
Because the problem will arise, I suppose, if you haven't got many females actually going on to trials, then you may not have enough to get the evidence from anyway, the outcomes.
I think sorry, Andrew, I don't know, maybe you want to answer that. I'm jumping in.
I would completely agree that you need data to kind of inform any decisions from that perspective. I think it depends on the compounds. They'll look at a lot of the what's called pharmacokinetics. So look at the dosing or the levels of the drug between different patients right at the very start. So if, for example, we find that there's a big disparity, particularly weight is a big one, they might dose by weight rather than a one fixed dose. So it just depends on the particular compound as to exactly, and whether there is a difference between any different subgroups.
Yeah, you've actually hit the nail on the head there. So when I asked this question 7 years ago, when I wasn't on a trial, but I just asked how the doses were decided on, and it said it was weight. And it immediately crossed my mind that although physiologically there can be similarities in weight, men and women are very different physiologically. But it seemed to work for me, so there we go.
Thank you, Lisa. We've got any other questions? Well, I just wanted to ask, thinking about you talking about comparing the model assisted and the model based designs. I guess it's sort of an opposite scenario to what you've seen in ProMMise. So if you had a dose that you were currently testing at a dose level that was seen to be safe, with the model assisted dosage, you keep putting patients into that dose level. But I was thinking about if you had some doses where the toxicity rates were quite close together, could the model assisted designs prevent you from exploring some of those higher doses, which may be associated with increased efficacy, because you just consider each dose in isolation?
Yes, it's two. I'll try to, obviously we're both statisticians, so I'll try and give an answer that's not too statistical. I think one of the problems with the model assisted is they tend to get towards the MTD and then flip. So if there's a little bit of a gap, you tend to get to one and then it recommends de-escalating. And the problem is with bigger sample sizes, it flips between the two, which is seen as perhaps a problem. It's in terms of the kind of the model based. It really does depend in terms of how you set the model up, as you know, and how sensitive you are within a particular region. So you can set it up to be quite sensitive, but then it doesn't give good operator, or it's poor when there's a bigger gap, for example, between the doses. So I think, there's kind of, as I say, I don't think it's a simple one or the other. I think there's pros and cons for both. And it's actually that the model assisted tend to do very well over a really big sample size, because they continue to collect information at doses that are close to the MTD, whereas the model based sometimes just recommend staying at one dose and don't explore if it's not been set up properly.
So I suppose if you were designing a trial and you had you thought that might be those sorts of scenarios may well play out in your trial, that we need to undertake lots of simulations. So when we're designing phase 1 trials, we as statisticians, we always talk about the amount of simulations that we're going to do in the different scenarios that we're going to do to explore what might happen. And I think that's a really essential part of phase 1 trial design, to see what might happen under different scenarios with different operating characteristics, and that's really part of the the need to really spend time on evaluating our designs at phase 1. Again, coming back to that efficiency, so that we're confident that the design that we end up with can answer the question that we want to answer. Okay. Anybody else got any questions or comments? No? Okay. So I'd like to thank Andrew for a very informative presentation. Really, really nice to see all of those designs summarised for us so well. I'd like to thank everybody for joining us for the webinar today. As I mentioned earlier on, but I forgot while we're recording, so for those who are watching this, we do have another schedule of webinars that are coming through, hoping to start the next round in November, and in the new year start to present some of the results from the trials that are currently going through final analysis within the CARP portfolio. So please do keep an eye out on the website and through Myeloma UK as well. So thank you very much everyone for joining. I hope you have a lovely afternoon, and hope to see you again soon. Thank you.
Thank you.